welcome to the deep dive. Today we are taking a plunge into a world you experience every single second of your life, yet which is largely invisible, silent, and mostly subconscious. The constant hidden language of human body odor.
It really is fascinating, isn't it? It's perhaps the most ubiquitous form of communication that we've been systematically trained, both scientifically and culturally, to ignore or even actively suppress.
Yeah, I mean, think about it. We put so much effort into deodorizing, masking, getting rid of our natural scent profile.
Yeah, absolutely do.
And that speaks directly to this long-held belief, both in science and just, popular culture, the idea that humans are microsmatic.
Right, the idea that we have a poor sense of smell.
Exactly, that compared to dogs or rodents, we're essentially scent-blind primates. Like, we traded away our powerful sense of smell for better vision.
That's right. And the scientific community, for decades, really largely accepted this idea, this evolutionary trade-off. But our sources today, they really demonstrate that this notion is fundamentally flawed.
So our mission today is to really dig into that, to comprehensively refute this microsmatic myth and uncover just how much complex, vital information you are constantly broadcasting and receiving through these chemosignals every single day.
We're talking about a chemical blueprint, one that carries data about your individuality, your health, your emotional state, even potentially your fitness as a mate.
Right. And to do that, we've analyzed sources covering, the hard science of chemical analysis, the psychology of things like competition and personality, and even the timeline of human sociality right from the moment we're born.
Exactly. But before we unpack all the specific data, especially when we get into social dynamics, competition, status, that sort of thing, there's a foundational concept we really need to define. It features heavily in our sources.
Okay.
And that concept is dominance.
All right, lay the foundation for us. What's the working definition of dominance that applies here in the context of chemical communication?
Okay, so dominance in the behavioral ecology sense that our sources use, it's not just, a personality trait like being bossy.
Right.
It's defined as an attribute of a pattern, specifically a pattern of repeated agonistic interactions.
Agonistic, meaning competitive, conflict-based.
Exactly. Competitive or conflict-based interactions between two individuals. And the key is that these interactions have a consistent outcome.
Okay, so it's not just a one-off fight.
No. One individual consistently wins, becoming the dominant partner, and the other consistently yields, becoming subordinate. It's about that established history.
Got it. So it's about a history of wins and losses establishing a relationship, not just someone acting powerful in the moment.
Precisely. It's the consistent behavioral outcome established through conflict. And this consistent status, once it's established, is something that other animals, and potentially humans too, need to be able to detect quickly, efficiently, to navigate their social world.
Makes sense. If you can tell someone's just lost a fight or has a history of losing.
Or conversely, a history of winning, you adjust your behavior accordingly. You don't necessarily have to fight them yourself to figure it out.
So the scent could be a shortcut to reading that social history.
That's the hypothesis. That scent communicates the status. It really frames a lot of how we think chemical signals might work in social species.
That immediately gives us a powerful lens. How can a chemical signal possibly communicate something as complex as a sustained pattern of wins and losses? We'll be looking for clues later, maybe in hormones or stress profiles, perhaps.
Exactly. We'll circle back to that when we look at competition. But first, let's really tackle this legacy idea that we're poor sniffers. As you said, for decades, the consensus was, primates, humans especially, traded smell for sight, better color vision, trichromatic vision.
And there was some biological evidence that seemed to back that up, right? Something about genes.
Yes, exactly. Molecular biology showed that primates have significantly fewer functional olfactory receptor genes, or R genes, compared to other mammals, compared to rodents or dogs, the classic good smellers.
So fewer genes coding for the smell detectors.
Logically seemed to mean weaker detection ability, fewer tools for the job. It looked like pretty conclusive evidence that our sense of smell was sort of on the evolutionary decline. An afterthought.
But that neat story is falling apart now. Recent science throws a wrench in that simple trade-off.
A massive wrench, yes. It's strongly challenged. While, the reduction in the number of functional OR genes isn't disputed, that's real, recent behavioral evidence tells a different story. It suggests the human olfactory system, the whole thing, including the main olfactory bulb in the brain, is actually far more sensitive and, frankly, more robust than we ever give it credit for.
More sensitive? How so?
Well, studies have actually found that for certain specific odors, primates, including humans, can be more sensitive than animals typically considered macrosmatic, like rats.
Really? More sensitive for some smells?
Yes. So it's not just about the raw number of gene types. It's about the whole system, how the receptors are organized, how the brain processes the signals. It's much more complex. The takeaway is that smell is far more essential, far more sensitive, and just everywhere in human biology than we've acknowledged.
Okay, myth busted. We're not scent blind. So let's move to the mechanism. If we're constantly broadcasting these signals, what's physically generating this chemical fingerprint? Where do these complex odors actually come from?
Right, so the odor profile, it's basically a mix. It comes from secretions from three main types of glands on your skin surface. And crucially, it involves the bacteria living on your skin.
Okay, 3 glands plus bacteria. Let's start with the most common gland.
That would be the eccrine glands. These are everywhere, all over your body. They produce this clear, watery secretion, mostly water, salt.
That's just regular sweat for cooling down.
Primarily, yes, thermoregulation. Because it's so watery, it's not the main source of that really distinct body odor smell, but it does provide the moisture, the environment for everything else.
Got it. Gland type 2.
Then you have the sebaceous glands. These are usually linked to hair follicles. They produce sebum.
That oily, waxy stuff.
Exactly. Protects the skin and hair. Sebum itself, it contains lipids, fatty acids. It can contribute to odor as it breaks down or oxidizes. But again, it's often more of an ingredient in the final mix.
Okay, so eccrine is watery, sebaceous is oily, which brings us to the main player for that characteristic human scent, the apocrine glands.
Precisely. The apocrine glands, these are really the major source of what we typically think of as BO or body odor, but their distribution is limited.
Where are they found?
Mostly concentrated in the axillae, the armpits, and the pubogenital area, the perianal region. And interestingly, they're basically dormant until puberty. They only become functional then.
And this is where the critical partnership comes in, right? You said bacteria are involved. Their secretions aren't smelly on their own.
That is the absolute key step. Apocrine secretions themselves are quite viscous. They're rich in proteins, lipids, steroids. But initially, they're pretty much odorless.
So what happens?
It's the action of the bacteria living on your skin flora. Primarily types like micrococci and diphtheroids. They metabolize these apocrine secretions. They break them down.
They eke the secretions.
In a manner of speaking, yes. And this bacterial breakdown produces the small, volatile organic molecules that we actually detect as odorants.
Can you give us an example? You mentioned one earlier.
The classic one is isovaleric acid. It's a common breakdown product, and it has that distinct, sometimes sharp, maybe pungent, even cheesy sweat-like odor. That's often a big part of what we perceive as intense body odor.
So when we say someone smells, what we're really smelling is the metabolic waste products of their skin bacteria.
Essentially, yes. It's the complex output of their unique resident bacterial community feeding on their apocrine secretions. It's like a little ecosystem on your skin producing your unique scent signature.
That totally reframes body odor. It's not just us, it's us plus our bacterial entourage. Okay, so these volatile compounds get released. How do we detect them? You mentioned 2 pathways. The main one is obvious, but tell us about both.
Right. So the primary one is the main olfactory system, MOS. This uses the olfactory sensory neurons, the OSNs, in the lining of your nose, the nasal epithelium.
The standard nose-to-brain pathway?
Pretty much. These neurons send signals directly to the main olfactory bulb, or MOB, in the brain. And this system is incredibly powerful. We have something like 350 different types of functional receptors, maybe more. And with those, we can detect and discriminate literally millions of different odorants, environmental smells, food smells, and most of that complex body odor profile we just talked about.
Okay, that's the main highway. Then there's the other one, the more mysterious accessory olfactory system, AOS.
Yes, the AOS. This one is traditionally associated with the vomeronasal organ, the VNO. You might have heard of it as Jacobson's organ.
Right. And snakes and mice, that's the pheromone detector, isn't it?
Exactly. In many non-human mammals, it's thought to be specialized for detecting pheromones, those specific chemical signals that trigger innate behaviors. Often it detects less volatile chemicals, sometimes requiring direct contact.
But the big debate is the VNO actually functional in adult humans?
That has been a huge debate for decades, and the current thinking. While the structure, the VNO, is present during human fetal development, and we do express some VNO-type receptor genes in our nasal lining, The prevailing view today is that the VNO itself is largely non-functional in adult humans.
The plumbing might be there, but the wiring's disconnected.
That's a good way to put it. It seems vestigial in adults.
Okay, so if the dedicated pheromone organ isn't working, does that kill the idea of human pheromones? Or does it just mean our main nose is doing double duty?
It strongly suggests the latter, which is actually fascinating. The sources point towards the idea that if pheromones, or maybe more broadly specialized chemical social signals, are important in human behavior and the evidence suggests they are, then the detection is probably handled primarily by the main olfactory bulb, MOB.
So our main smell system just picked up the job.
It seems likely it incorporated the ability to detect these key social signals. They aren't processed through a separate, dedicated channel. They're integrated right into our main sense of smell.
Which might explain why these signals often feel subconscious. We don't consciously register pheromone detected. We just feel an attraction or a slight repulsion or perceive something as pleasant or intense.
Precisely. It's all part of one integrated system, constantly analyzing every volatile molecule floating around us, whether it's from coffee or flowers or the person sitting next to you.
Okay, so that unified system is clearly doing way more than just checking if the milk's gone bad. It sounds like it's running a constant, subtle background check on everyone we meet. If we're broadcasting all this unique info, then smell must act like a specific, stable chemical fingerprint.
Absolutely. And this is where the research moves beyond just asking people, does this smell nice? Or do these smell different? It gets into the hard chemistry, identifying the actual molecules involved.
And the key method for that seems to be GCMS analysis, gas chromatography, mass spectrometry. You mentioned this was used in a big study in the Austrian Alps. For listeners not familiar, why is GCMS so crucial for this kind of work?
Right. GCMS is really the gold standard here. It does two critical things. First, the gas chromatograph, GC, takes a complex mixture like sweat and separates it out into its individual chemical components, usually based on things like boiling point and how they interact with the column inside the machine.
So it sorts the chemicals.
It sorts them. Then the mass spectrometer, MS, takes each separated component and basically weighs it very precisely, breaking it down and creating a unique mass signature, like a chemical fingerprint for that specific molecule.
So you can identify what each component is.
Exactly. It's incredibly sensitive. It can detect and identify hundreds of different volatile and semi-volatile molecules, even in tiny amounts, all from one sample, which is exactly what you need to deconstruct something as complex as human body odor.
And this big Austrian Alps study, with nearly 200 people, sampled repeatedly over 10 weeks. What did this powerful technique reveal about our sweat?
Well, first, it confirmed where the main signal is coming from. Axillary sweat, armpit sweat, was significantly richer in these volatile compounds than saliva or urine.
So the armpit is really the main broadcasting station.
It seems to be a major one, yes. On average, they found over 240 distinct chemical peaks, distinct compounds in a single sweat sample. But here's the crucial part for the fingerprint idea. Across all the samples, they identified 373 peaks that showed remarkable intra-individual stability.
Whoa, 373 stable peaks within one person. So over 10 weeks, despite diet changes, stress, whatever, those specific compounds stayed consistent for that individual.
That's right. That stability is the biological basis for a chemical fingerprint. It's reliable over time for that person. And from these stable compounds, the researchers went further and actually identified the chemical structures of 44 specific individual marker compounds, molecules that were highly characteristic of particular individuals.
44 markers. But you said earlier, it's not about finding one unique gene-DOE molecule, right? It's the overall picture.
Exactly. That was a really crucial methodological finding. Successful individual identification, making a positive ID based on smell. It's almost never about finding one single unique compound that only that person has.
So how does it work then?
It relies on analyzing the pattern of the entire volatile profile, looking at the qualitative differences, which molecules are present or absent, but maybe even more importantly, the quantitative differences.
The amounts.
Yes, the relative ratios and concentrations of those hundreds of stable compounds. You know, person A might have a lot of compound X and just a little of compound Y, while person B has the opposite ratio. That unique pattern, that specific blend, that's your chemical ID badge.
That's incredibly complex.
Yeah.
Moving from the individual signature,
Okay.
what about group differences? How does smell communicate something like gender?
Well, for gender, the perceptual cues, what people report smelling, are often probabilistic and tied strongly to intensity and pleasantness.
Right.
Meaning, generally, if human raters smell a sample and judge it as stronger and less pleasant, they are more likely to assign it to the male category. And that happens somewhat regardless of the actual sex of the person who provided the sample.
Is that just a cultural bias, like men are smellier?
Yeah.
Or is there a biological reason behind that perception?
There seems to be a strong biological link. On average, men tend to have physically larger apocrine glands than women.
The glands that feed the bacteria.
Exactly. Larger glands mean potentially more substrate for the bacteria to work on, which can lead to a higher concentration of those volatile byproducts. This correlates directly with perceived odor intensity.
So intensity becomes a proxy cue for maleness.
It seems so. In fact, the studies showed that if you took a sample from a female donor that happened to be particularly intense, raters were highly likely to misclassify it as male simply based on that intensity cue.
Interesting. But the chemical analysis, the GCMS, did it find specific chemical markers for gender, beyond just overall intensity?
Yes, it did. The Austrian study identified 12 marker compounds that were statistically characteristic of gender. They broke it down further. 7 markers were more characteristic of males.
Like what? Any examples?
Sure, things like pentadecanoic acid and hexadecanoic acid. These are saturated fatty acids. And for females, they identified 5 characteristic compounds, for instance, isopropyl hexadecanoate.
Okay, so subtle chemical differences are there. It's not just one big male smell molecule. It really reinforces that idea that our noses, or our olfactory system, must be incredibly good at picking up on these complex patterns, these combinations of chemicals.
Absolutely. It's clearly processing multivariate information. It's a spectrum, not an on-off switch. Which actually leads us nicely to the next category, age, and the famous or maybe infamous concept of the old person odor.
Right, this is something people talk about anecdotally all the time. Do the studies actually confirm that we can reliably smell someone's age group?
They absolutely do. There was a key study where researchers collected body odor samples, typically from pads worn in the armpit, from 3 distinct age groups. Young adults, 20-30, middle-aged adults, 45-55, and old-age adults, 75-95.
And could people tell them apart just by smelling the pads?
Yes. Raters could discriminate between the age groups significantly better than chance. And critically, they were particularly good at correctly identifying the samples from the old age group. They could assign that old age label much more accurately than they could for the young or middle age groups.
So the smell of the oldest group is the most distinct, the easiest to pick out.
It seems to be the most salient age-related odor signal, yes.
Okay, but here comes the big surprise, the paradox. Most people, if you ask them, would probably describe the smell of old age as strong, maybe unpleasant. But the data from the study completely flipped that expectation.
It is totally counterintuitive, based on common assumptions. The sources clearly found that the body odors collected from the old age group, 75 to 95, were rated as less intense and also less unpleasant compared to the odors from both the young and the middle age groups.
Less intense and less unpleasant.
Wow.
Which group smelled the worst then, according to the raters?
Actually, odors from the middle age group, 45-55, were rated as the most intense overall and also generally the most unpleasant. And interestingly, the odor from the old male group was rated as the least intense and least unpleasant of all categories.
So the stereotypical old person smell isn't actually strong or bad, according to the science. It's detectable, it's distinct, but it's relatively mild and not particularly unpleasant. So why? Why would our noses be tuned to detect this specific milder signal? This brings us back to that evolutionary speculation you mentioned earlier, the good genes idea.
Exactly. It poses a fascinating evolutionary question. Why retain the ability to detect an odor signal that indicates someone is past their peak reproductive years? The argument, the speculation, centers on what reaching a very old age actually signifies.
Meaning survival itself is a signal.
Precisely. An individual who has successfully survived to, say, 80 or 90 years old has clearly demonstrated extraordinary adaptive advantages throughout their life. Most notably, they must possess a robust, highly effective immune system to have weathered decades of exposure to pathogens and environmental challenges.
So the subtle scent of old age isn't necessarily a signal of decay or weakness, but rather a signal of proven resilience, a testament to high immune competency. Like a Lifetime Achievement Award in survival coded in scent.
That's the thinking. Signals indicating old age might be subtly detected and not perceived as repulsive because they indirectly hint at good genes, specifically genes conferring longevity and immune strength.
So even if you're not directly mating with that elderly person, knowing that their genetic line produces individuals capable of reaching that age could still be valuable information, in a broader evolutionary context, maybe influencing mate choice within that family line.
It's possible. The immune strength, communicated chemically, could still be seen as an indicator of underlying genetic quality. It completely reframes the perception of the smell of age not as something negative, but as a specific, subtle, potentially high-value signal of biological resilience.
Okay, so this hidden chemical dialogue starts right from birth and shapes our social world. Let's trace that timeline. How do these odors organize our relationships? Starting right at the beginning, the mother-infant bond.
Olfaction is critical right from moment one. It develops incredibly rapidly. Newborns show an innate attraction to the odor of milk and specifically breast odors.
That makes sense biologically, guides them to the food source.
Exactly. It's a crucial adaptation for survival. Volatiles released from the mother's areola region, possibly combined with slight changes in pH there, act as key chemical cues that help guide the baby to latch on correctly.
And the ability to recognize their own mother's scent develops quickly too.
Astonishingly quickly. Studies have shown that within just a few days, infants can reliably distinguish their own mother's breast odor from that of a stranger's. And this preference, this ability, actually gets sharper. It significantly increases between just 2 and 10 days of age.
Wow, that fast. So those early close physical interactions are constantly reinforcing this olfactory link.
Absolutely. It provides constant opportunities for mutual kin olfactory labeling. The infant learns the mother's unique chemical signature, and you can bet the mother is also learning the baby's specific scent. It forms the basis for rapid subconscious identification and is a key part of that early bonding process.
It establishes that first fundamental olfactory relationship, the biological bedrock of attachment. Okay, so the child grows up, eventually reaching puberty, and the olfactory system seems to shift gears, preparing for reproductive roles. This brings us to that fascinating, if slightly controversial, idea about inbreeding avoidance. How might smell help steer relatives away from each other reproductively?
Right. As hormone levels surge during adolescence, the function of the olfactory system does seem to shift, becoming more attuned to reproductive signals. The hypothesis, and it's still debated, is that around puberty, close genetic kin, like a daughter and father or a sister and brother living together, might start to develop a kind of mutual olfactory aversion.
Aversion. They start to dislike each other's smell.
Or at least find it less pleasant than the smell of unrelated individuals. It's thought to be a subtle, evolved chemical mechanism. The olfactory system recognizes the scent as kind, based perhaps on shared MHC profiles or other genetic markers, and this recognition then generates a subtle hedonic penalty. The smell just isn't perceived as attractive.
So evolution basically installed a subconscious chemical stop sign to reduce the likelihood of attraction between close relatives during their peak reproductive years.
That's the idea, an unconscious safeguard promoting outbreeding. And it aligns perfectly with how the major histocompatibility complex, the MHC, which we'll get to, seems to work in mate choice, generally favoring partners who smell different or dissimilar. It's all about ensuring genetic mixing and fitness.
Subconscious biology guiding fundamental social rules. Amazing. Okay, let's move into more general adult social interactions. We tend to think we navigate these using sight and sound conversation. But the handshake study suggests there's a whole lot of silent chemical investigation going on, too.
Oh, the handshake study is just brilliant. It's such elegant experimental evidence. It proves that even these really common formalized social rituals, like shaking hands, are actually conduits for transferring chemical information.
How do they prove that?
They showed that the simple act of handshaking effectively transfers specific volatile compounds from the skin of one person to the skin of the other.
What kinds of compounds did they identify them?
They did. They found several molecules commonly present in sweat and sebum being transferred, including things like geranyl acetone, squalene, and hexadecanoic acid. Just clasping hands is enough to leave this chemical residue from the donor on the recipient's hand.
Okay, so the transfer happens, but what's the behavioral consequence? Does the person who received the handshake do anything with that chemical information?
Yes, that's the kicker. The recipient's behavior changes immediately afterwards in a very specific and measurable way. The frequency and the duration of that person touching their own face, particularly near their nose, increases significantly after shaking hands compared to before.
They sniff their hand more after shaking someone else's.
Essentially, yes. It's interpreted as a subconscious chemical investigation of the person they just interacted with. You shake hands, you acquire this little chemical data packet on your hand, and then without even thinking about it, you bring your hand closer to your nose to covertly sample the information you just picked up.
That's incredible. We think we're being all formal and polite with a handshake, but seconds later, we're unconsciously sniffing our fingers to get the real story.
It seems to be a very primal drive, just masked as a subtle, maybe self-soothing or grooming-like behavior. And the sources even mention a really powerful observation related to this, involving individuals with autism spectrum disorder, ASD.
Oh, tell us about that. What do they observe?
Well, when they conducted similar handshake experiments with a group of cognitively able adult men with ASD, some of these individuals showed behavior that was much more obvious. Instead of the subtle, fleeting face-nose touching seen in the neurotypical population, some participants with ASD exhibited really quite overt, sometimes persistent, hand-sniffing behavior after the handshake.
So the subconscious behavior became conscious and explicit.
In some cases, yes, it highlights how fundamental this olfactory investigation might be. Perhaps for individuals who process social cues differently or lack certain typical social inhibitions, the olfactory channel becomes a much more dominant and explicit way of gathering information about a new social partner. It sort of reveals the underlying drive when the usual social filter isn't fully engaged.
Fascinating. And that leads perfectly into the adult interaction, perhaps most governed by chemical signals.
Right.
Choosing a mate, and the role of the major histocompatibility complex, the MHC.
Right, the MHC. This is a really critical set of genes. It's technically a large genomic region containing a set of highly polymorphic genes.
Polymorphic meaning they vary a lot between different people.
Exactly. Huge variation across the population. And these genes are absolutely vital for the adaptive immune system. They code for proteins, cell surface receptors mostly, that are involved in recognizing foreign invaders like bits of bacteria or viruses and presenting them to T cells to initiate an immune response.
Okay, so having diverse MHC genes means your immune system can recognize and fight off a wider range of pathogens. Makes sense. But how on earth does your body odor signal what your MHC genes are?
That's the million-dollar question, and the exact mechanism isn't fully nailed down. But the prevailing theory involves metabolites. The idea is that the specific metabolic byproducts related to your immune system activity and just your general metabolism, which is influenced by MHC, are released in sweat and other bodily secretions.
And these metabolites are volatile. They create a smell.
Some of them are, yes. Or they interact with skin bacteria to produce volatile compounds. These act as subtle chemical markers that reflect your underlying MHC genetic profile. And the really compelling finding, replicated in many studies across different species, including humans, is that individuals generally show a preference for the body odor of potential partners who have complementary or dissimilar MHC genes compared to their own.
Complementary meaning different. Why is different better?
We seem hardwired to seek out dissimilarity in MHC. If your MHC profile is, say, type A, you tend to find the scent of someone with type B more attractive than another type A. The evolutionary logic is clear. Mating with someone who has different MHC genes maximizes the genetic diversity at these crucial immune loci in your offspring.
So your kids inherit a wider range of immune defenses.
Exactly. They get a broader toolkit to fight off more types of infections. It increases their chances of survival. So this preference for dissimilar MHC-related odors is thought to be an evolved mechanism to ensure outbreeding and boost the immune fitness of the next generation. And critically, who you find attractive based on scent will vary depending on your own specific MHC makeup. It's a personalized preference system.
Okay, so we've covered the stable signals, who you are, your age, your genetic compatibility. Now let's get into the dynamic stuff, the immediate signals. What happens when we're under pressure? How's our internal state, like winning, losing, feeling stressed, broadcast through our scent? The study on mixed martial arts fighters seems like the perfect arena to test this.
Yes, the MMA study was really well designed for this question. It looked directly at how the outcome of a physical competition, winning versus losing, affected the perceived pleasantness, the hedonic quality of male body odor.
How did they set it up?
They recruited 32 amateur male MMA fighters. They collected body odor samples using pads worn under the arms before the match, that's the baseline, and then again immediately after the match finished. They also measured the fighter's mood or affective state and got saliva samples to measure hormones like testosterone T and cortisol C.
Okay, first things first. Did the sheer physical act of competing change the smell, regardless of who won or lost? Were there universal effects?
Oh, absolutely. The physical competition itself had dramatic and consistent effects on the odor profile. After the fight, regardless of outcome, the perceived pleasantness and attractiveness of the body odor significantly decreased, and the perceived intensity of the odor significantly increased.
So fighting just makes you smell worse, basically.
Yeah.
More intense, less appealing.
That's what the raters perceived, yes. The intense physical exertion and presumably the emotional stress of the competition made the resulting scent universally less attractive and stronger smelling.
Interestingly, though, they also found that perceived masculinity of the odor increased after the fight.
They did, yes.
Yes.
Which is fascinating. Even as the overall pleasantness dropped, something about the physiological state post-fight chemically boosted the perception of masculinity. So it confirms the act of intense physical competition itself fundamentally alters how body odor is perceived. It's a massive physiological and endocrine event.
Speaking of endocrine events, what about the hormones? Cortisol, the stress hormone that must have gone up.
Definitely. Cortisol levels were significantly higher in the post-match samples compared to the baseline across the board. This confirms the fight was a highly stressful, high-arousal event for everyone involved, winners and losers alike.
Okay, that makes sense. But the finding on testosterone T was a bit different, wasn't it? It didn't follow the expected pattern.
That's right. This specific study found no significant effect of the competition outcome, winning versus losing, on testosterone levels. This was somewhat surprising because it runs counter to the predictions of the challenge hypothesis in some contexts.
The challenge hypothesis being the idea that winners get a T boost and losers see a drop, reflecting the change in social status.
Generally, yes. That's often observed.
So why do you think they didn't find that T effect here? Was it just this group of fighters, or the timing, or something else?
It's a great question, and the paper discusses possibilities. It could be several things. Maybe the T response in amateur fighters is more variable or takes longer to manifest than the immediate post-fight sample captured. Or perhaps the sheer intensity of the stress response, the massive cortisol spike seen in everyone, might have overwhelmed or masked any subtle outcome-dependent T changes in that immediate time frame.
So the overwhelming signal right after the fight was just stress, cortisol, maybe drowning out the I-1 lost testosterone signal, hormonally, at least initially.
That's a plausible interpretation of these particular findings, yes.
Yeah.
The stress was universal and acute.
Okay, so stress levels were high for everyone. But did the outcome winning versus losing have a specific measurable effect on the smell itself? Did losers smell different from winners?
They did, and this connects the emotional state directly to the scent. While the statistical significance was described as borderline, the trend was clear. Losing the match affected the pleasantness of the body odor more profoundly than winning did. The smell of defeat seemed to carry a distinct, more negative chemical signature that the raters were picking up on.
Wow. And this is where the path analysis comes in, right?
Yeah.
They didn't just show a correlation, they tried to show a causal link between the emotion and the smell. Tell us about that mechanism.
Yes, path analysis is a statistical technique that lets you test proposed causal relationships between variables. And what their analysis showed was really critical. The link between losing the match and having less pleasant body odor wasn't direct. It was mediated by the fighter's negative affective states.
Mediated? Meaning what, exactly?
It means the unpleasant odor wasn't just a direct result of the physical act of losing. The odor quality changed because losing the fight made the fighter feel bad. It induced negative emotions, things like feeling distressed, maybe ashamed, unhappy.
So the chain reaction was lose fight, right echo, release specific chemicals, smell less pleasant.
That's exactly the pathway the analysis supported. The emotional response to the loss was the crucial intermediate step that altered the chemical signal.
We literally smell the emotional consequences of social defeat. The body broadcasts its distress or shame through chemicals and sweat that other people can detect. That's an incredibly honest signal. You can't easily fake that.
It really is. It's biological proof that our emotions aren't just internal feelings locked in our heads. They are physical, chemical events that manifest outwardly on our skin, potentially for others to perceive. And evolutionarily, that makes sense, right? Knowing if a rival is distressed might signal weakness. Knowing if a potential partner or ally is distressed might signal a need for support or intervention.
Okay, so that's communication based on immediate state winning, losing, stress. What about more stable long-term personality traits? Can we actually smell if someone is, say, generally dominant or typically anxious?
Another fascinating study tackled exactly that. It linked volunteers' self-assessed personality traits using the standard Big Five model, plus they added dominance as a separate trait to how judges rated their body odor. People wore t-shirts, the shirts were collected, and then raters smelled the shirts and tried to guess the personality of the wearer.
Just based on the smell of the t-shirt. Okay, which personality traits could people guess most accurately?
The results were really interesting. The traits that showed the highest correlations, the ones judged most accurately from body odor alone, were precisely the ones most linked to emotionality and social arousal. Extroversion had a correlation of R equals 0.36, neuroticism was R equal 0.34, and dominance was R equal 0.29.
Extroversion, neuroticism, and dominance. That's pretty remarkable accuracy just from scent. What about the other big five traits?
Judgments for the others, agreeableness, conscientiousness, and openness to experience were much weaker, mostly non-significant. The signal seems strongest for those traits related to emotional expression and social interaction styles.
The fact that neuroticism is chemically detectable makes a lot of sense, given what we just discussed about stress and negative effect.
Absolutely. Neuroticism is characterized by a tendency towards negative emotions, anxiety, stress sensitivity. It aligns perfectly with other research showing people can sometimes smell fear or acute anxiety. When someone's anxious, specific stress-related compounds are released in apocrine sweat. So it stands to reason that someone who is chronically anxious, a hallmark of high neuroticism, might consistently emit a subtle chemical signature related to that state.
Like a constant low-level anxiety flag in their scent profile. And conversely, extroversion. That's linked more to positive emotionality, sociability.
Exactly. Extroversion is associated with approach behavior, social assertiveness, maybe higher positive arousal. This might relate to different underlying hormonal profiles, or perhaps a distinct metabolic signature that produces specific, maybe more neutral or even pleasant volatiles, especially during social engagement.
And finally, let's revisit dominance. Why was that trait accurately judged, especially considering it's about social interactions? And wasn't there a specific nuance about who was doing the judging?
Yes, the accuracy for judging dominance from scent was particularly high when the participants were rating donors of the opposite sex.
Ah, the mating context again.
The researchers suggested exactly that. It implies that the chemical signal communicating dominance, or perhaps a lack thereof, is especially relevant and maybe more easily detected when assessing potential mates.
So when you're evaluating someone as a potential partner, assessing their social standing or competitive ability becomes really important, and scent gives you this immediate, maybe unfiltered clue.
That's the interpretation. It's a quick, potentially subconscious assessment of their social viability. Interestingly, the studies also noted that the ability to judge dominance accurately seemed to correlate positively with the rater's own self-assessed extroversion.
So more extroverted, maybe more assertive people are better at picking up dominance cues in other scents.
It suggests that, yes, perhaps due to a shared sensitivity, maybe linked to neurophysiology or hormones, that makes them more attuned to those specific social signals. Now, realizing that body odor carries all this information, identity, genetics, emotion, personality, it obviously has huge practical implications, especially in the field of medicine.
Yeah, it's easy to forget that connecting smell to health isn't some new age idea. It's ancient. Doctors used to rely on it, right?
Absolutely ancient. We're talking Hippocrates back around 460 BC. Physicians in ancient Greece and Rome, and through the Middle Ages, figures like Galenus, Avicenna, they explicitly used the smell of the patient's body, their breath, their urine, their feces, as fundamental diagnostic clues.
So does the breath smell fruity? Wasn't just a random question.
Not at all. Specific smells were associated with specific ailments. Of course, modern technology, high-tech imaging, blood tests largely replaced diagnosis by smell, but the underlying principle holds. Illness does change body chemistry, and some of those changes produce volatile compounds that can be smelled. Specific volatiles released by sick patients can act as key indicators of disease.
And now we're coming full circle, using modern science to harness that principle. What are the applications today?
There's intense research focus now on developing odor-based diagnostics. Probably the most famous recent example, though anecdotal, is Joy Mill.
The woman who could smell Parkinson's disease.
That's right. She's a Scottish woman, apparently a super smeller, who reported being able to detect a distinct musky odor on her husband many years, potentially a decade, before he was formally diagnosed with Parkinson's.
That's just incredible. The power of the human nose.
It really is. And scientists are trying to translate that natural ability into technology. Because of evidence from super smellers like her, and also from trained animals like dogs who can detect certain cancers, Researchers are developing highly sensitive tools.
Like electronic noses.
Exactly. Electronic noses, or e-noses. These are arrays of sophisticated chemical sensors designed to detect specific patterns of volatile organic compounds, VOCs, that are known markers for certain conditions. Things like melanoma, lung cancer, colorectal cancer, or neurodegenerative diseases like Parkinson's or Alzheimer's. The goal is often early, non-invasive detection.
Amazing potential. Now, let's circle back quickly to a chemical signal we talked about earlier, cortisol, the stress hormone. You mentioned this cortisol paradox, that some studies found high cortisol was actually linked to more attractive body odor. That seems totally backwards. Why would we be attracted to the smell of stress?
It is a challenging adaptive puzzle, absolutely, because normal high chronic cortisol isn't great. It's a glucocorticoid, and these hormones can suppress the immune system if levels are persistently high.
Right. So why the attraction?
The hypothesis, the best guess explanation, is rooted back in adaptive mate choice and immune signaling. It suggested that perhaps only individuals with really robust, strong immune systems, highly immunocompetent individuals, can actually sustain high cortisol levels, maybe due to a high-stress lifestyle or environment, without getting sick or significantly compromising their overall health.
Ah, okay. So if you can handle high stress, evidenced by high cortisol, and still smell good or function well, it implies your underlying immune system must be exceptionally strong to cope with that hormonal load.
That's the idea. The preference for C-associated odor, in this view, becomes adaptive because it's indirectly signaling a robust immune reserve. The scent essentially says, I'm under stress, but my system can handle it, I'm still thriving. And that signal of underlying health and resilience, communicated through the odor, becomes desirable in a potential mate, overriding the simple negative connotation of stress.
Fascinating. Okay, so olfaction is clearly vital for social processing, maybe health assessment. What happens when this crucial system breaks down? How do we classify smell disorders?
Right. Smell disorders, broadly termed dysosmias, represent some kind of alteration in the sense of smell. They're generally categorized into quantitative and qualitative forms.
Okay, starting with quantitative, that's about sensitivity, how much you can smell.
Exactly. Quantitative disorders are about changes in sensitivity. You have anosmia, which is the total, complete loss of the sense of smell. This can be incredibly disruptive to quality of life, impacting taste, hazard detection, social connection. Then there's hyposmia, which is a partial loss or reduced sensitivity. This is actually very common, especially as people get older. And much more rarely, you can have hyperosmia, which is hypersensitivity and abnormally acute sense of smell. This is quite rare and often linked to specific neurological issues or hormonal changes.
Okay, so that's loss or gain of sensitivity. What about qualitative changes, where the smell itself is wrong?
Right. Qualitative disorders involve distortion or illusion. Parosmia is probably the most commonly discussed one recently. That's where a familiar smell is perceived as something different and usually unpleasant. So coffee might suddenly smell like garbage, or flowers might smell like burning chemicals. This is often reported by people recovering from viral infections that damage the olfactory system. And then there's phantasmia, which is smelling something that isn't actually there. An olfactory hallucination or illusion. People might report smelling smoke or sewage or something metallic when there's no external source.
We've certainly heard a lot more about smell loss and distortion recently. What are the main things that cause these dysfunctions?
The underlying cause is usually some kind of damage or degeneration affecting the olfactory system, often starting with the olfactory epithelium, the smell-detecting tissue high up in the nose. The major culprits include head trauma, chronic sinus infections, rhinosinusitis, nasal polyps, exposure to certain toxins, neurodegenerative diseases, and very prominently in recent years, viral infections.
Yeah, COVID-19 really brought olfactory dysfunction into the spotlight for millions.
Absolutely. Viral infections, whether it's SARS-CoV-2 or even just a severe common cold or flu, are a major cause of post-viral olfactory loss or distortion. And we can't forget aging. Age-related decline in smell, presbyosmia, or just age-related hyposmia is very common. It's estimated to start subtly even in the 5th decade of life in healthy people and tends to progress gradually.
Are there factors that make recovery less likely if you do lose your sense of smell?
Yes, there are negative prognostic factors. Generally, the more severe the initial loss, the harder the recovery. The longer the symptoms have persisted before seeking treatment, the worse the outlook. Advanced age at onset, especially being over 75, is also linked to poorer recovery. And lifestyle factors like smoking can also hinder recovery.
Okay, so if someone does experience smell loss, say after a virus or head injury, is there anything they can do? Are there proven treatments, especially non-drug options?
Fortunately, yes. There is one leading evidence-based non-pharmaceutical intervention that has shown real promise. It's called olfactory training.
Olfactory training, like physiotherapy for your nose.
That's a great analogy. It's essentially a structured program of repeated, deliberate sniffing designed to stimulate and hopefully rehabilitate the olfactory system. It's simple, non-invasive, and patients can typically do it at home.
How does it work? What do you actually do?
The standard, well-studied protocol involves consistent practice, usually 12 weeks of training performed twice every day. The patient actively sniffs 4 specific, distinct, high-intensity odors. The classic quartet of odors used covers different scent categories: phenylethyl alcohol, which smells like roses; eucalyptus, camphoraceous, medicinal; citronelle, lemony, citrusy; and eugenol, clove-like, spicy.
So you just sit and sniff these 4 things twice a day for 3 months.
Pretty much. For about 10-20 seconds per scent. It's not just passive exposure. You're meant to concentrate on the smell, try to recall what it should smell like. It's like focused neurological exercise.
And does it work? What's the success rate?
It's shown significant success. This repeated, short-term, focused exposure seems to encourage neuroplasticity, the brain's ability to reorganize itself. It can promote structural and functional changes in the olfactory pathways. Studies indicate that this kind of training regimen improves olfactory performance, sensitivity, and identification in roughly 25% to 33% of patients suffering from post-traumatic, post-viral, or idiopathic unknown cause smell loss.
So maybe 1 in 3 or 1 in 4 people see real improvement. That's actually quite hopeful.
It is. It's solid evidence that the olfactory system isn't static. It can be trained and potentially recover function, even after damage or in adulthood.
This has been a truly fascinating deep dive. It feels like we've completely overturned that old idea of humans being microsmatic, having a poor sense of smell. That view just seems definitively revised by all this research.
Absolutely. The body of work we've discussed really paints a picture of human olfaction as this incredibly sophisticated, high-bandwidth communication system, constantly running, mostly beneath our conscious radar.
So to synthesize the key takeaway for you, the listener, your body odor is anything but random noise or something just to be washed away. It's a complex chemical fingerprint.
Right. It encodes rich information about your individuality. Remember those 373 stable chemical peaks. It signals your sex, often through intensity, but also specific markers. It conveys your age with that distinct, subtle profile of older individuals. It hints at your genetic compatibility through MHC dissimilarity cues.
And maybe most surprisingly, it broadcasts your immediate internal emotional states, like the smell of defeat mediated by negative feelings and even stable personality traits like neuroticism, extroversion, and dominance. It's like a chemical ID badge and mood ring rolled into one.
It informs everything from kin recognition and bonding, potentially inbreeding avoidance, assessing social status, right through to mate selection. And the value of digging into this system scientifically is huge. That combination of high-tech chemical analysis like GCMS with clever psychological testing is unlocking a really fundamental aspect of human biology.
From developing potential diagnostic tools like e-noses for catching diseases like Parkinson's or cancer earlier, based on their scent signature,
to understanding the deep biological roots of social dynamics, like how emotional states directly translate into detectable chemical signals. The science really confirms that scent is this powerful and often surprisingly honest communication channel. Those correlations linking personality traits to scent. They're often as strong, sometimes even stronger, than correlations found when people judge those same traits just from seeing a face or hearing someone speak briefly for the first time.
which brings us right back to that cultural contradiction we started with.
Right.
You mentioned the sources note this research is often done in Western industrialized societies.
Yes, and a defining feature of many of these societies is that we are increasingly systematically deodorized. Think about it. Intensive daily washing, powerful antiperspirants designed to block sweat, deodorants to mask smell, and just the pervasive use of perfumes, colognes, scented lotions, laundry detergents.
We surround ourselves with artificial scents and actively try to eliminate our natural ones.
Exactly. These practices introduce a huge amount of chemical noise, and they actively mask or even obliterate our natural body odors.
So here we have this incredibly sophisticated biological system honed over millennia of evolution to transmit and receive critical information for survival, social bonding, reproduction. And culturally, we're doing everything we can to mute it, to turn down the volume, or just change the channel completely.
Which leads us to the final provocative question, the thought to leave you with. What might be the real functional consequences, socially, reproductively, maybe even physiologically, of this widespread aggressive detachment from our natural chemical communication system?
What happens when we silence that signal?
By constantly adding strong chemical noise with artificial fragrances, or by completely silencing the natural signal with antiperspirants, are we inadvertently messing with crucial biological processes? Maybe hindering subtle aspects of social bonding, disrupting those evolved cues for kin recognition or MHC-based mate assessment? Are we even making it harder to subconsciously gauge the health or stress levels, or maybe even the emotional honesty of the people around us?
It creates a weird feedback loop, doesn't it? The better we get at masking our real scent, the more we have to rely only on sight and sound, maybe pushing that subtle, honest chemical language further into the shadows.
We might be trading a degree of chemical honesty and biological signaling for social conformity or aesthetic preference, and we really don't yet understand the full biological or social cost of that ongoing exchange.
Something profound to think about next time you reach for that extra strength antiperspirant. Thank you for joining us on this deep dive into the hidden language of human body odor.
Thanks for listening.
We'll see you next time on The Deep Dive.